In this video SOP, we'll take a look at the ABEC 300-liter bioreactor and provide guidelines for basic operation of the 300-liter fermenters at the ABPDU. Each ABEC reactor is on a skid that includes the reactor, the utilities, and the control computer with a human-machine interface. Proper in-person training is required before using this equipment independently, but this video will serve as an introduction and reference for general ABEC procedures. Safety. Use of the 300-liter ABEC bioreactors is a high-risk operation because of the materials used, heat, pressure, and scale of the reactor and auxiliary devices. Not following safety instructions can lead to serious injury to yourself or others. As with any process in the lab, proper PPE should be worn when setting up and operating the ABEC bioreactors. One of the major risks associated with the ABEC reactors is the large quantities of acid and base used for pH control. Direct contact with these corrosive materials can cause burns or skin irritation, and their fumes can cause headaches, eye irritation, or respiratory distress. Given these risks, pre-assembled and pressure-rated tubing is used for these assemblies. Other measures in place to prevent acid-base related safety incidents include securing all connections before any procedure is run on the vessel, clamping the assembly tubing while setting up, refilling, or moving containers around, marking the hang tag after each use, and pumping at a low speed. Once the line is locked into the pump, the other clamps can be removed. Another hazard associated with operating the ABEC reactors is the hot surfaces created by continuous steam use. Steam lines run throughout the ABEC skid, through the pipes surrounding the bioreactor, and around the addition port connections. When steam is actively running through these lines, a high temperature sign should be posted on the reactor and caution should be taken when using hand valves. Steam, along with the air and water, also contribute to pressure hazards. All these utilities are provided at around 30 psi and the vessel is pressurized during operation. Even when not being operated, residual pressures in the vessel and lines can create a hazard when opening valves, filters, or removing any piece from the head plate. Always check the pressure gauge on the top of the reactor and manually vent if necessary by opening XV03 until pressure gauges read 0. Given the scale of the reactor, the energy requirements are also significant. Shock or arc flash from a 480-volt source can cause serious injury or death. For general operation, it is critical that lab operators have completed the requisite electrical switching training and wear appropriate personal protective equipment while powering up the reactor skid. The 480-volt cabinet powering the skid creates a shock hazard when opened. As such, only qualified electrical workers are able to work inside for any reason. In case of an emergency, there is an emergency power off, or E-stop, on the main control panel of the reactors. The E-stop will shut valves, turn off the agitator, and put the reactor in an idle state until the emergency status is lifted. Utilities. The ABEC fermenter is turned on using the electrical switch on the side of the control panel. This powers low voltage equipment like the computer controls, as well as high voltage equipment like the agitator motor. Once the equipment is on, we can open the utility shutoff valves located on the back of the skid. Chilled water is sent to the heat exchanger and condenser for cooling. Planned steam provides heat for temperature control. Clean steam is used for sterilization-in-place, or SIP. Clean dry air is used for pneumatic valve operation and is process air which is supplied to the reactor during operation. And nitrogen is sparged to get a 0 point for the dissolved oxygen calibration. The operating pressure for all utilities should be regulated between 25 and 35 psi. One exception is clean dry air, for which the additional connection at 80 psi is required. Equipment inspection. Before we can proceed, we need to take some time to visually inspect the reactor skid. Start by checking utility pressure gauges for correct working pressures. Take a look at the outside of the reactor to spot any irregularities like loose connections from a previous run or dismantled parts, and make sure the interior is clean with no signs of deterioration. Check the head plate. The sight glass is removed often during preparation and cleanup. Both the hand valves and pneumatic valves on addition ports should be in the closed position, and probe ports should be sealed. As a final check, there should not be any residual pressure on the vessel. Cartridge filter installation. There are 3 filters on the 300-liter fermenter: the exhaust filter, the sparge filter, and the overlay filter. Each filter requires a cartridge filter and a housing, which needs to be installed as part of the reactor prep. Remember to always make sure there is no pressure in the vessel before opening any part of the fermenter or skid. We'll start by installing the exhaust filter located at the top of the front of the skid. This filter filters outgoing air from the reactor to avoid contaminating the lab with any biomaterial. To start, remove the clamp and cap, then screw the filter cartridge in place. Next, secure the housing over the cartridge. It's important that the exhaust housing has a relief valve so back pressure can be released manually in case the filter gets clogged during a foam over event. Replace the tri-clamp and ensure the relief valve is closed. The sparge filter filters inlet air being fed through the sparge line underneath the agitator, and it's the lower of the 2 filters on the back of the skid. The process for installing the filter is the same. Screw in the filter, add housing, and secure the clamp. The overlay filter filters gases being fed into the headspace and is just above the sparge filter. Repeat the installation process a 3rd time. Functionality tests. To test the general reactor functions, put the reactor into run mode. After running a couple of minutes and checking for leaks at the heat exchangers and valves, return the reactor to idle mode. Another functionality test that will be performed multiple times during reactor setup is a pressure test, or P-test, to check the ability of the vessel to hold pressure. To perform a P-test, remove all forces on the process synoptic screen. This sets all valves into a neutral position and allows the reactor controls to open and close valves as required by the procedure. Go to the current procedure window and select Start P-test. A prompt will appear to ensure all addition ports are closed. Use this time to confirm the vessel is sealed, all tri-clamps and hand valves are tightened, and a high pressure sign has been hung up. Process air is pumped into the vessel until the pressure is about 16 psi. It will hold this pressure for 10 minutes to confirm there are no leaks. It will then depressurize safely. If the P-test passes, move on to the next step. If it fails, troubleshoot by checking valves, filter housings, tri-clamps, and rerun the P-test. Another important procedure to execute as part of the functionality checks is the sterilization-in-place procedure. The cycle is performed before the media is made up to test heating and cooling functionality of the reactor and provide an estimate of how much condensate is added during an SIP cycle. This information is used later when batching the media. As always, check the residual pressure before opening the reactor through the head plate, using XVO3 to release pressure if necessary. Fill the reactor with water to the target media weight. Close the vessel and rerun the p-test to test the head plate seal. After the vessel passes the p-test, record the pre-SIP vessel weight and move to the SIP menu using the HMI. The purpose of SIP is to bring the temperature of the interior of the vessel to 121 degrees Celsius. Initiate the procedure by pressing the start button and watch the reactor going through the multi-step process, one step after the other. The reactor jacket is drained of any water to prepare for plant steam injection during the heating stages. During the Heat 1 heating stage, the temperature of the vessel is increased from room temperature to approximately 100 degrees C by continually running plant steam through the reactor jacket and by injecting clean steam through various ports into the reactor. Direct clean steam injection allows for a more rapid temperature increase, but it comes at the expense of significant accumulation of condensate, which needs to be accounted for during the batching process. Once the Heat 1 temperature set point is reached, the exhaust valve closes. Pressure gradually increases to facilitate temperatures greater than 100 degrees C. Heating continues until the vessel temperature is equal to the SIP set point of 121 degrees Celsius. The vessel temperature is at 121 degrees Celsius. The reactor enters dwell mode, a 30-minute holding period during which the temperature is maintained constant. Once the sterilization time is up, the air inside the reactor is replaced by directing clean process air through the overlay filter, and any heating is discontinued. The reactor is cooled down to 95 degrees Celsius during the Heat 1 by pumping cooling water through the jacket. Pressure is maintained above ambient to avoid the liquid inside the reactor from boiling. Cooling continues to the SIP exit temperature and the exhaust valve opens to vent pressure. SIP cycle concludes and the reactor enters growth mode. Record the weight after SIP to see how much condensate was added during the SIP process. This is usually around 20 kilograms. Probe installation. pH probes must be calibrated once prior to being put in the reactor using standard buffers and once after sterilization using a media sample. Start by inspecting the probe for damage or debris, then connect the probe to the reactor. Enter calibration mode by pressing Cal on the pH transmitter. Select 2-point as the type of calibration, and place the probe in the pH 7 buffer. Rinse with DI water and shake dry. Press enter. Once the signal stabilizes, the transmitter will automatically display a prompt to place the probe in the second buffer. Remove the probe, rinse with DI water, dry, and place the probe in the second buffer. press enter. Once the reading stabilizes, the display will automatically show the slope calibration factor S and offset calibration factor Z. Select Yes to save calibration. And press enter. The reactor must be drained before installing the probes. To drain the tank, put the reactor into idle mode and vent to take any residual pressure off of the vessel. Connect a hose to the 1.5-inch tri-clamp fitting at the harvest port XV19 and route the hose to a floor drain. Open the XV19 port on the control screen and wait for the water to drain. Once the tank is empty and there's no pressure on it, you can install the pH and DO probes. Replace the blanks at the bottom rear of the vessel with the probes. It is good practice to have a second pH probe installed, although not calibrated, in case of a loss of function on the original probe in the middle of the run. The DO probe is installed without being calibrated. The DO probe also requires a signal converter from digital to analog. Plug the reactor line into the converter and the converter line into the probe. One final inspection, ensuring the transmitter lines and the probes are secured, is always good. Sterile assemblies. Sterile assembly vessels are used for corrective agents like antifoam, inoculum, acids and bases, or any media component that must be sterilized separately. For each assembly, use a length of appropriately sized tubing made of a compatible material long enough to run from the assembly vessel through the pump and to the desired additive port on the fermenter. Here's a quick guide on how to set up sterile assembly bottles. Attach a dip tube to the bottom port of the additive bottle top or cap. Next, attach a 0.2 micron filter to the non-dip tube outlet and the transfer line to the dip tube outlet. A hand valve goes on the other side of the transfer line. Make sure this hand valve is closed. A red band marks the position of the hand valve. The red tape is visible when the valve is open. During assembly, pay close attention to the orientation of the hand valve. If installed incorrectly, the assembly is compromised and will not work as intended. Connect the barb to the hand valve using a tri-clamp and attach the transfer line. Secure all connections with zip ties as shown in the example. It is good practice to label the container with date, purpose, project ID, and initials. Sterile assemblies can come in many different sizes, but the components and assembly procedure largely stays the same. Note: pre-assembled and pressure-rated tubing sets are used for sterile assemblies that hold corrosive materials, like acids and bases. A bioburden assembly is used to collect a sterile media sample to test sterility before running the experiment. For this assembly, you will need a plastic bottle attached to a sample port with an air filter. Once complete, the assemblies then go into the autoclave for sterilization before they are ready for use. To install the bioburden assembly, remove the sample port that is on the vessel. Replace the sample port with the autoclaved bioburden assembly. Ensure clamps are tightened. To sterilize the bioburden port, open the steam valves above and below the sample port and let sit for 15 to 20 minutes. Caution: this will be hot. To install sterile addition assemblies, connect the sterilized hand valve to the reactor by first removing the hand valve or caps that are in place in the desired port and replacing them with the assembly hand valve. Once the line is locked into the pump, clamps can be removed. Perform a p-test once everything is installed to test these new components: the pH probes, the sterile addition assemblies, and connections. Batching. Before media components can be added to the reactor, the load cell that is used to track the vessel weight needs to be taired. Enter the growth mode and prime the jacket to get the correct tear weight. Tear the load cell and return the reactor to idle mode. Dry chemicals and water are added through the addition port on the head plate. Remember, whenever opening the vessel, to check the pressure. Add enough roti water to immerse the bottom impeller to ensure proper mixing. Then add the dry chemicals, using a funnel if necessary. Rinse and adjust to the target weight pre-SIP as specified in the batch record. When batching is complete, replace the sight glass and secure tightly. Perform another p-test to ensure the head plate is sealed again. Sterilization in place. Once the media components have been batched, the second SIP takes place. This SIP is meant to sterilize the media at the target vessel weight. The SIP procedure is the same. Before starting the procedure, be sure the high pressure and high temperature signage is posted, and record the pre-SIP weight. Again, the vessel will move through 2 heating stages, a dwell period, and 2 cooling phases. Once the sterilization is complete, record the post-SIP weight. Post-sterile additions. With the autoclave solutions, route the tubing from the sterile addition assembly through the pump being used for the PSAs. Install the assembly following the same procedure as before. To sterilize the port, go to the Addition Port Control window on the HMI. Find the port that the new assembly is on and manually open the 2 valves as shown to start the port sterilization. After 20 minutes, press the Remove Forces button to shut the valves and stop the sterilization procedure. Allow for at least 5 to 10 minutes to cool. Open the hand valve on the assembly, then open the corresponding port on the HMI. Next, enable local pump control by turning on the pump. Use the dial on the pump to adjust the speed to a safe operating set point. It's recommended to start the pump at 2.1 and slowly increase the speed once the transfer has started. Flip the switch to the left to start pumping. The solution is now being transferred into the reactor through a sterile connection. Once the addition is complete, stop the pump by flipping the switch back into the neutral position. Remove all forces on the HMI. This shuts the addition port and disables the pump. Close the hand valve. Heat-sensitive solutions are passed through a 0.2 micron cartridge filter for sterilization instead of autoclaving. To use the cartridge filter, displace the air and wet the filter by starting the pump with the vent open. Once water comes out of the vent port, close the vent and transfer the solution into the reactor. Turn off the pump, close the valves, and remove the assembly as needed once complete. The growth media is now complete. Pre-inoculation tasks. Base priming. In order to prime the baseline, remove the clamp from the outlet line, open the hand valve, and on the HMI, open the drain valve. Again, set the pump no higher than 2.1 and turn on. Watch the see-through hose barb until you see base come through. Turn off the pump and tear the base scale. Turn on pH control and adjust pH to an intermediate value below the final pH target to leave room for error. Work in small increments to avoid overshooting on the way to the desired pH. For checking the growth media's sterility, take a sample by opening the sample port on the vessel, then open the hand valve on the bioburn assembly and aseptically fill the plastic bottle with media. Close the sample port and the hand valve, then remove and transfer the whole of the bioburn assembly to the nearest biosafety cabinet. Filter the media using a sterile 0.2 micron vacuum filtration assembly. Once the solution has been filtered, remove the lid from around the filter and use sterile tweezers to transfer the filter paper to a petri dish face down. Incubate the petri dish at 37 degrees C. The flow through from the bioburden sample can be used for an offline pH measurement. Compare the benchtop pH meter result with the online pH reading on the bioreactor. If there is a significant difference, recalibrate the bioreactor pH probe. Run through the same pH probe calibration procedure outlined before, but select Process as the type of calibration and enter the offline pH value. Record and save the new calibration. With confidence in the pH signal, the pH is adjusted to the final target. DO probe calibration. For the DO probe, turn on the nitrogen sparge and wait until dissolved oxygen levels stabilize. Calibrate 0 point to set the DO reading to 0%. Turn off nitrogen, turn on air sparge, and wait until the liquid is fully saturated with air, indicated by a stable DO reading. Calibrate slope to adjust the reading to 100%. Record and save the calibration. Note that the DO probe calibration should be carried out using the same conditions for temperature, agitation, pressure, and air sparge as for the experiment. Auxiliary devices. Auxiliary devices such as feed pumps, external scales, gas analyzers, and computers with customized software applications are used to provide additional control capabilities required to execute complex fed batch fermentation processes. In the following, we see the initiation of the data acquisition software and the setup of the feed. To set up the feed, move the holding tank with the feed onto the floor scale behind the reactor. Run the tubing through the green pump designated as the feed pump. Then attach the hand valve outlet to the feed addition port as was done with the other assemblies. Inoculation. Take the sea culture grown in the shake flasks out of the incubator and place them into the biosafety cabinet. Check for contamination and transfer the required amount aseptically into a sterile assembly bottle. Move the inoculum over to the reactor and steam on the assembly as you would another sterile addition. While the port sterilization procedure is ongoing, it is time to turn on control loops, such as DO cascade and pH control. To enable off-gas measurements, open the valve of the exhaust line as shown. Once ready for inoculation, open the addition port and hand valve. Start the pump and transfer the inoculum into the reactor. The inoculation marks the beginning of the experiment. To collect a sample, open the sample port valve on the reactor, followed by the hand valve on the sample assembly. Collect as much sample as is outlined in the experiment sampling procedure, then close the hand valve, followed by the reactor valve. Sterilize the port by opening the steam line hand valves above and below the sample port. Open the hand valve momentarily to clear the line and steam the outlet. The experiment is in progress and continues until the end of fermentation. Harvest. The standard downstream process used at the ABPDU is the disc stacked centrifuge, which separates the fermentation broth into cells and supernatant. Attach the drain port of the reactor to the inlet port of the DSC. Open the drain port on the HMI and run the DSC according to its SOP. The harvest is over when the reactor is empty. Reactor cleaning. To clean the reactor, add water and carry out a pressure test, followed by an SIP cycle to heat kill any biological material that remains in the reactor. After draining the reactor, we can begin setting up the vessel for cleaning in place, or CIP. For CIP, both the probes and cartridge filters should be removed and capped. Replace the sample port assembly with a CIP connector piece. Open the sample port valve and all the hand valves as shown in the video to enable the flow of cleaning fluids. The reactor is now in the cleaning configuration and the CIP wash cycle can begin. Once the CIP cycle is complete, the reactor is clean and ready for the next experiment.